This will be the second part of our discussion on nucleic acids. The title slide just kind of gives you an overview of the differences between RNA and DNA. And so RNA is shown on the left here. Okay, it's single-stranded, and the bases that you see in RNA are C, G, A, and U. DNA, on the other hand, is double-stranded, and the bases are C, G, A, and T. So T is used in DNA, and U is used in RNA. Otherwise, the bases are similar. Right, so today we're going to be finishing up Chapter 8, and we're going to be working through Section 3, dealing with the chemistry of nucleic acids, and then a little bit about, in Section 4, it's a smaller section of other functions of nucleotides. But first, we're going to review something we sort of talked about at the end of the lecture last time, and that was the discovery of DNA. And this was the DNA X-ray diffraction pattern. Okay, this was found by Rosalind Franklin in the early 50s, and her advisor, Maurice Wilkins, suggested to do this experiment, and Rosalind Franklin did. Okay, this pattern suggested from this pattern that DNA was in a helical structure. It had two periodicities in it, 3.4 angstroms and 34 angstroms. It didn't show this cell in this crystal structure diffraction pattern did not show anything about a double helix. Okay, and it was really Watson and Crick that came up with this idea of a double helix. How they came to that is they put Chargraff's work in Chargraff, if you remember, that was the work that said the amount of the base A in DNA is equal to the amount of T, and the amount of G equals the amount of C. So they kind of put together that A has to pair with T and G has to pair with C, along with this helix, and they came up with the double helix. And an anti-parallel double helix would fit the data. Okay, how they came to this, though, Wilkins showed this data that Rosalind Franklin got to Watson and Crick without her knowledge. So a lot of, I guess in lore, it sort of says it was stolen. I mean, it wasn't really stolen per se, but that data was really kind of shared without her knowledge. Okay. And again, like I mentioned, Watson, Crick, and Wilkins shared the 1962 Nobel Prize for this. Franklin could not share in it, though, however, because she had died in 1958 before that. But you can also see how important this was. Watson and Crick came up with this idea in 1953, and less than 10 years later, they were winning the Nobel Prize, which is a relatively short time between publishing something, discovering something, and getting a Nobel Prize. Okay. Well, hydrogen bonding is really important in DNA, and we'll go through this in a little bit more detail. So these pictures we'll look at in a little bit larger size here. I should probably use my highlighter. There we go. So these pictures we'll look at in a little bit larger size here in a second. But basically, the hydrogen bonding interactions in DNA, two bases hydrogen bond to form a base pair. When these nucleotides are monomers, you can have a large number of base pairings possible, but in a polynucleotide chain, nucleic acid chain, there's only very smaller number of hydrogen bonding pairs possible. Right? What we refer to as Watson-Crick base pairing, those are going to be really the majority of the base pairings in double-stranded DNA, and those would be where A pairs with T and G pairs with C, as shown on the right here. And another way you can say this is that purines are going to pair with pyrimidine, or A purine will pair with A pyrimidine. So this is the Watson and Crick model. Again, this is an anti-parallel double helix. That means one strand is running one direction and the other strand runs the other direction. We'll see a better picture of that in a second. There's hydrogen bonding between the two strands, and that occurs between the bases and the strands. And then the bases are also stacked, and that's shown here in these yellow stick figures. You can see that they're really kind of stacked on top of each other. Okay, and probably better, easier to see that in the space filling model here. The yellow are the bases, and you can see that they're stacked really well on top of one another. Okay. So looking at it, these hydrogen bonding pairs in a little bit more detail, right? Well, G-guanine is pairing with cytosine. They make 3 hydrogen bonding interactions, and those are shown. Okay, the ketone group, the carbonyl group on guanine will pair with this amine group on cytosine. This sort of in-chain amine group will pair with the nitrogen in the ring on cytosine, and then the amine group on guanine will pair with the carbonyl on cytosine. So that's a kind of a pattern here. You have a carbonyl hydrogen bonding with an amine group on a different base. Okay, so that's seen twice in this GC pair. It's also shown once here in the AT pair, where the amine group on adenine hydrogen bonds with the carbonyl group on thymine. And then you also have this in-ring NH and N hydrogen bonding pair. Okay. So these are the hydrogen bonding interactions that you should be really aware of and familiar with. DNA strands, though, when it makes a double helix, the strands are referred to as complementary. And that means that the sequence on one of the strands is complementary to the other, meaning that they're not the same sequence, but they're going to have the bases that will hydrogen bond with the bases in the first strand. Okay. And again, as I mentioned, the sequence is going to differ between both strands, and it's read just by convention. It's read from what's known as the 5' end to the 3' end. And remember that 5' and 3', those numbers come from the ribose sugar. Okay, so the 5' phosphate that's in the ribose and the 3' OH group. Okay, so on one strand it'll go 5' to 3' in this direction. The complementary strand is read 5' to 3' in the opposite direction. Okay, and that's what we mean when we say these are anti-parallel. Okay, so this really when Watson and Crick came up with this model, they really suggested that this is a way that it can be replicated, right? The two strands can separate, and then each strand can be read and replicated individually. And that actually, it turns out how this occurs. And in next semester, you'll get into more of the specifics on that. Okay, one strand is synthesized, pretty much all continuously, and the other strand, because of the directionality, has to be synthesized in chunks, and then those are sort of connected back together. Okay, but that, again, that's next semester, not going to be important in this class. Okay, so when you replicate the genetic code, first you have to have the strand has to separate, these two strands, so the blue strands have to separate from one another. Then each strand will serve as a template. Okay, so we refer to those as parent strands, and then the DNA that is synthesized from those we refer to as the daughter strands. Okay. This synthesis is catalyzed by enzymes known as DNA polymerases. You'll talk more about these next semester. Okay. the newly made DNA is going to have one strand that's a parent strand and one strand that's a daughter strand. So you'll have two copies, two double-stranded copies of the DNA from a single double-stranded parent copy. The backbone in DNA is really actually pretty flexible, right? There's lots of single bonds in here if we look at this. So this would be the backbone. And really they're all single bonds going through this, which means they can all rotate, okay? And maybe this double bond here in the ring structure, that's probably adds a little bit of rigidity. But other than that, these are all single bonds that can rotate, okay? What that means is there's lots of options for bond angles, and you can get more DNA structures. Okay. Some other ways that DNA can have more structures are listed here, right? When you have 4 or more adenosine residues in a row, it can produce a bend, 18-degree bend. If you have a palindrome or inverted repeat sequences, they can form different kind of DNA structures. And then you can also get things known as hairpins or cruciform structures in DNA. So we'll see examples of these here in the next few slides. The take-home message is there's not one structure of DNA. It's a dynamic molecule, which is actually kind of important, because if it was more like a protein where there was one 3-dimensional shape of it, and maybe it has some movement to it, but it doesn't really adopt other totally different structures, it'd be very hard to actually unwind it and be able to replicate it and or express genes from it. So it is actually pretty important that it is somewhat flexible and dynamic. This is given from the book. It's three main forms of DNA. So one is called the A form, another is the B, and then lastly we have the Z form. Okay, and they're all a little bit different. A and B are closely related. They're both right-handed helices. The diameter is a little bit different, so it's almost like the A form is just a B form that has been kind of smashed down and flattened a little bit. And when you do that, the diameter is a little bit wider in the A form than the B form. The bases per turn a little bit more than the B form. The rise is a little bit less than the A form to the B form. And then the what's not shown in the table, the periodicity between turns here, that would be 28 angstroms in the A form, and that was 36 angstroms in the B form. So right, it's less than the B form. So it's almost like the B form got kind of smashed down. The Z form is completely different. The Z form is a left-handed helix. Some of these other numbers probably aren't very important to remember because, right, if I'm asking you about these structures and I say it's a left-handed helix of DNA, you know that from right there, it's going to be the Z form. Okay. Another nucleic acid molecule, all right, that was DNA. RNA is another type of nucleic acid molecule. Okay, and messenger RNA specifically is what carries the code for the sequence of proteins. So that carries this genetic code from the DNA to the ribosome to be translated into a protein. Okay. It's synthesized using the DNA template. The difference between RNA and DNA, the most striking difference is right there in the name. It contains ribose instead of deoxyribose. Right. Another difference, as we've mentioned, RNA contains uracil, the base uracil, and that replaces the base thymine. One messenger RNA can code for more than one protein, and we'll see an example of that and define a term for that here in a second. But that's also very kind of interesting, if you will. And RNA, messenger RNA, it works together with transfer RNA or tRNA, and that really transfers this genetic information. And really, I think the better term for that is translates that genetic information from nucleic acid to amino acid, DNA to protein. And a structure of a small length of messenger RNA is shown here, and you can see that it sort of makes a helical structure, but it's single-stranded, and so these are a little bit more flexible still than DNA would be, because it is only one strand. Okay. The term for a messenger RNA that encodes only one protein, one gene that encodes one protein, is monocistronic. Okay. And the messenger RNA, again, is read from 5' to 3'. Okay. And there's usually some sequence on the 5' end and the 3' end that isn't translated. So those are shown here in gray. And the red part of this messenger RNA is what actually encodes for the protein. So that's referred to as the gene that encodes for the protein. Okay, so when the messenger RNA has one gene or one protein that it encodes, that's monocistronic. When there are multiple genes for multiple proteins, that term is that we use is polycistronic. Okay. The other sequences or other structures of DNA that can occur, or I should say nucleic acids, not just DNA, occur from palindromic sequences. Okay. And palindromes, if you don't know, palindrome, that term means a word or a phrase that are the same when they're read either backward or forward. Okay, and then the book gives some examples here. This Finnish word, if you read it, and I'm not even going to try to pronounce that, but if you read it one direction, it's the same one direction as the other direction. This Greek saying is like that too, but like if I read that backwards, doesn't look like what it says forward. So I suppose this is a lowercase letter for that. That's another example the book uses, but there are probably some better examples to use than these. All you need to know is if you have a DNA sequence that has palindromes in it, that is, it's the same forward as it is backwards, then you're going to get some superstructures formed. In particular, what are known as hairpins or cruciforms. Okay, so here we have an example of a palindrome. So if you read this sequence, at least this part that's highlighted of the sequence, TTAGCAC, right? And then if you read it the other direction on the other strand, that actually matches up with that. So what you can have there is, instead of base pairing normal this way, you can have some other funky base pairing between strands. Okay? Where actually now you can get base pairs formed between nucleotides that are on the same strand. Okay, and we'll see that in a second. And then you have these other repeats of sequences. Something like this would be termed a minor repeat, where you have a sequence shown here, TTAGCAC. If you have that same sequence, maybe just in a different direction, right next to it, you can get some interesting base pairing from that too. Right. So here we have this palindrome sequence if we have this sequence written, and then there's a few bases in between it, and then this sequence, right? These are complementary sequences, and they can form these base pairs with one another instead of the opposite strand. So now you have base pairing within the same strand, and this forms what's known as a hairpin. So a hairpin is when you have one strand that is hydrogen bonding with itself, and then the little loop up here where you don't have any sort of base pairing in there. These loops can be, a little bit bigger or smaller depending on where you have these complementary sequences, but that would be where the hairpin. Okay. In RNA molecules, because they're single-stranded, they don't have a complementary strand to hydrogen bond with. They're going to hydrogen bond with themselves, and they can form some very really complex sort of structures, and those are shown here in one example. Not going to need to know, and there's really no way to for you to be able to predict a structure like this on an exam. So really just kind of highlighting this as something that RNA does. Okay, it'll probably be a little bit more important when you go through the whole translation process. Okay, and some other sort of interesting things that can happen in RNA is that you can get hydrogen bonding between guanine and uracil, okay, that you don't really see in DNA, double-stranded DNA. You can see that kind of stuff in RNA because it is single-stranded. There's a little bit more degrees of freedom there. This is a transfer RNA structure, and this also goes to show that you can get some, somewhat unique, at least unique in terms with respect to DNA, hydrogen bonding in transfer RNA, and some of those are shown here, like cytosine and guanine forming hydrogen bonds together, and the adenine and uracil have sort of normal hydrogen bond pair, but then you can also have another adenine hydrogen bonding with the 2' OH group of uracil. And so those kind of hydrogen bonds are found in transfer RNA molecules, and that's what's depicted here. Okay, so this is a transfer RNA. Again, you'll see more. Probably see this again next semester. Okay. That brings us to the central dogma of molecular biology. In the central dogma, really states what molecules contain information that can be transferred to other molecules. Historically, it was said the central dogma was really that DNA, the information is stored in DNA that can be transcribed into RNA, and then RNA can be translated to protein. So that's kind of the information flow that you would see within living organisms. Okay, DNA to RNA to protein. And that's kind of where it started, but as we've gone on and researched more of this, since the 1950s, I supposed, Francis Crick really published this in 1970, but first proposed it in the late 50s. Okay, this central dogma idea. But since we've been, researching this for, going on, I guess that would be, something like 60 some years, there's been some other discoveries that have shown you can have DNA, right? Information from DNA going to DNA, that would be DNA replication. Okay, these dashed lines are filled in too from the subsequent work in the decades since then. You can have DNA information going to protein. at least in a test tube in a lab that has happened. Okay, you can have what's known as reverse transcription, where you have RNA molecule that's reverse transcribed into DNA, and retroviruses act in that way. Okay, so that's one way that information can go backwards. RNA can be replicated. So viral RNA is replicated. RNA silencing in eukaryotes, that is a relative recent discovery that shows that information, RNA, can be replicated. Okay, and so this is probably currently the most complete picture we have of the central dogma. And again, the process, you'll go through this more next semester, but the process of going from DNA to RNA, that's called transcription. So you can kind of think of that. These are both nucleic acids. They're both using a nucleic acid language, if you will. So you're just transcribing one to the other. Okay, RNA to protein, that's what we term translation. You're going from a nucleic acid language to an amino acid language, so different languages. Okay, so moving on to section 3, nucleic acid chemistry. Okay. And one thing that's really important in DNA, nucleic acids, is this denaturation. And that's where you have this double strands of a DNA separating. That's what we refer to as denaturation. Okay, the covalent bonds in the DNA remained intact, which is important because that sequence that's in that nucleic acid molecule remains the same. So the genetic code remains intact. Okay. What's being broken are the hydrogen bonds between the two strands are being broken, and the strands can separate. And when this happens, the UV absorbance increases because the bases, when they're in a double strand like this, are stacked, and that actually lowers the UV absorbance. And when they denature, the UV absorbance increases. Okay, so that's one way you can measure the denaturation of a DNA strand. Okay. And this denaturation is really thought of as being reversible. Okay, especially if you're using smaller sequences, you can think of it as being reversible. And the reverse process is called annealing, when two single strands of DNA come together in base pair and form a double strand. That's annealing. Okay. This is used in this process is used of denaturation and annealing is used in both DNA sequencing and in PCR, and that PCR stands for polymerase chain reaction. So if you do any work in a microbiology lab, you're going to use PCR. So the denaturation of DNA is affected by a couple things primarily. The base composition is important. Higher C and G content will increase the melting temperature. And remember, C and G form 3 hydrogen bonds to one another. A and T only form 2 hydrogen bonds. So the more G and C you have, the more hydrogen bonds you're going to have in your double strands, and the harder it's going to be to separate those two strands. Okay. And we refer to we call this a melting point, where the temperature where the strands sort of separate from one another, although it's not necessarily really melting, but that's what it's referred to. So the T sum M stands for melting point or melting temperature. And you would read that on a chart. Your chart would look something like this, where you have a certain absorbance and then UV absorbance, and then remember when the strands denature, that absorbance goes up, so it stays pretty constant until you reach a certain temperature. Strands start coming apart, and then after a while, the strands are fully apart, and this levels off to a maximum absorbance. Okay, and then that midpoint there in that region, this sigmoidal region, would be the melting temperature. If you're going to find that from data, you would do the derivative of that, and then it would be, the maximum point of that derivative. So that would be, basically in the middle of this curve, this changing part of the curve. Right. The melting point is going to depend on a couple things. One is the G and C content. The other most common thing it depends on is the length. So the longer your DNA strand is, the higher the melting temperature is going to be. And that's because the longer your strand is, the more hydrogen bonds you're going to have in that double strand. Okay. It really becomes, the length of this becomes much more important for shorter pieces of DNA. If you're looking at the change in melting point from a sequence that's 10 nucleotides long opposed to 15, that's going to have a much bigger difference than if you're looking at a piece that's 100 nucleotides long compared to something that's 105 nucleotides long. Okay, so that the length is going to be more important for smaller pieces. But you do use small pieces of DNA a lot of times in the lab, and those, what we refer to as primers, are usually fairly short. Okay. And then finally, the melting temperature is going to depend on pH of the solution and ionic strength. Salt, if you add a higher level of salt, that will actually increase the melting temperature. This one, at least in a laboratory setting, when you're doing work, microbiology work, you tend to be using commercial kits, and they don't really have, you're using standard solutions. So this one, you really don't vary the salt very much, at least when you're using, the typical commercial kits that are sold. What you really try to vary more is the sequences that you're using for your primers, and to do that you would either change the length of them, or maybe pick a different part of the DNA sequence that has higher GC content or lower GC content to try to get the correct melting temperature for your primers. This chart just shows you the percentage of G and C with respect, the melting temperature with respect to the percentage of G and C, and you can see it's a straight line, so they're linear. It's a linear relationship there. The higher percentage of G and C you have, the higher the melting temperature is going to be. Okay. And you can actually see how different regions of DNA can differ when you're melting it. So the AT regions, the regions that are higher in A and T, are going to be single-stranded, and that's where these circles are. These regions have higher A and T content. Okay, because those regions, localized regions, actually can melt before some of these other regions that have higher GC content. So that's what this picture's showing. An important way of working with DNA, we really need to know the sequence of DNA to be able to work with it. And the first method developed to sequence DNA was known as the Sanger method. Okay, and Sanger actually, not only did he develop this method for sequencing DNA, he also developed a method for sequencing proteins. So he has two methods to sequence these biomolecules. When you hear the term Sanger sequencing, that really is referring to the DNA sequencing, though. Okay, so Sanger sequencing, DNA sequencing, that was the first method for doing this. And how do you do it? Well, your contents in your sequencing reaction, you're going to have your DNA of unknown sequence, or you can refer to this as your template sequence. That's let's say you're trying to sequence the genome of some organism, that would be the DNA from your sample. Okay. Then you have what's known as a primer. We're not You're not going to really be since we're not actually going through how this works specifically, at least in this chapter, you probably don't need to know all these terms, but it will become more important, especially next semester. But your DNA primer is a short sequence that matches up to a particular part of your target DNA, your template DNA. Okay. And the DNA primer is needed because for a DNA polymerase to work, you need this primer to be there to match up to the template and tell the polymerase where to start adding sequences. And so the polymerase uses the template strand, and it basically copies that strand, but it puts the complementary base in to the template. Okay. And so this would be the primer, this would be your template, and your polymerase would be adding bases down the chain. Okay. Then you also need deoxynucleotide triphosphates, and those are the monomers that you need to propagate the chain. And you also include what are known as dideoxynucleotide triphosphates. So those are nucleotides that have the deoxynucleotide. Remember that has one of the OH groups missing, the 2' OH group missing on the ribose. The dideoxy has the 2' and 3' missing. So when one of these nucleotides is added to this chain, this OH group is no longer there, and so there's nowhere to keep that chain going. There's nowhere to stick that 5' phosphate from the next base onto it. So when you have that, the chain terminates. Okay, so it's important to have these dideoxynucleotides, A, G, C, and T, in equal quantities to one another. And so here's a picture of one of those dideoxynucleotides. So the DDNTP is dideoxynucleotide triphosphate. Okay, so you have no OH groups there on that ribose ring. Okay, so the old way of doing this is you add these things together. You do your PCR polymerase chain reaction, and you make certain sequences, and right, so you're copying your template, and most of the time it's going to add one base in each of these. There's a chance that it could add a dideoxy base at any point here and stop the chain. So you're going to get pieces that you amplify that are for each of these lengths, one nucleotide more. Okay. And then you separate via a gel. You can separate in 2 dimensions, one based on if the last base in the chain that you've made is either an A, a C, a G, or a T. And then you can separate them in a second dimension based on their size, right? So you get up here, you would have 12 nucleotides long. Then the next line would be for 11, 10, 9, 8, and so forth. And from that, then you could read a sequence. Okay, so you could read the sequence if you're doing it in the 3' to 5' direction, right? The first base here would be A, second base would be G, T, C, and so forth. Okay, so that's sort of an older method of doing it. Right, more recently, again, you use the same technique where you have a template and a primer, and you have 4 deoxynucleotide triphosphates and 4 dideoxynucleotide triphosphates. You do a polymerase reaction. At every point in that chain, there's a chance that it could be a dideoxy that's added, and each of these has a separate fluorescent label on them. Okay, you then can put those through a gel electrophoresis instrument, and measure the absorbance for this label that is individual for unique to each individual base, and you get a graph that looks like this. Okay, and depending on the color there of the peak, so each peak that comes out represents a different product, a different length product that's formed. So the shorter ones come out first, the longer ones, come out later. And so each step here represents one more nucleotide on your product, and you can tell by the color of them which nucleotide it is, so then you can get a sequence from that. Okay. So this would be the more, I guess, what's the term I'm looking for? More modern way to do Sanger sequencing. But I should mention too that now there's what's termed as next-gen sequencing or next-generation sequencing, which can actually go through the sequence of a genome much quicker. This method was used for the first genotyping, the first discovery of the human genotype, and it took many years to get through that together. But now with next gen sequencing, you can get the same amount of information in a day. So it's sequencing, genetic sequencing has really come a long way. Well, DNA can it's important to know the sequence of DNA. Sometimes you get what are known as mutations in DNA, and that's what can lead to some problems, right? And a mutation is just when a base is changed, a nucleotide is changed in your DNA sequence, and then your genetic code it gets altered. And there's a few different mechanisms for mutations. Okay, and they're shown here. We have 5 of them that we'll talk about in the class. One is deamination, 2 depurination. So deamination, you're losing an amine group. Depurination, you're losing a whole purine base. Ionizing radiation, alkylating agents. Okay, those are things that stick hydrocarbons to your bases and change them that way chemically. And then finally, reactive oxygen damage, and also referred to as ROS, okay, for reactive oxygen species. So that's kind of might seem a little striking that there's so many ways that your DNA can be damaged and ultimately mutated, but cells have mechanisms to correct most of these modifications. So the first one is deamination, and the molecular mechanism for this is chemical. It's not like something where an enzyme catalyzes this reaction. It happens spontaneously, just via a chemical process. Okay, and so the examples shown here are, any base that has an amine group in it is susceptible to this, where you have your amine group, your NH2, that just gets replaced with an O carbonyl group. So if you had that, like say in cytosine, this amine group, if we replace that with a carbonyl group, now that's uracil. So deamination of cytosine will lead to uracil. Okay, 5-methylcytosine is one of those sort of uncommon bases we talked about, where the 5 position is methylated. If you have the deamination of 5-methylcytosine, that gives you the same, what would be 5-methyl-uracil, which is just thymine. Okay. Adenine, when you lose that, you get a carbonyl here, and that's what we refer to as hypoxanthine. Okay, that's not one that you have to memorize the structure of. Okay, and then finally, guanine also has an NH2 group, a amine group, and when it loses that, it forms what's known as xanthine. Okay. For the cytosine deamination, that occurs about 100 times a day per cell. So if you're looking at a given cell, that's going to happen 100 times per day in that cell. Okay. What that does, this is probably the most important one here, because that changes a C to a U. Okay, so the what happens if there's a mechanism that if you have a U in your DNA sequence, there's an enzyme that recognizes that and says, oh, that was a mistake. I can fix that. Okay, and so that's why it's important that we have the base T, thymine, in DNA as opposed to uracil. Okay, so if uracil was present in DNA, there'd be no mechanism for repairing the deamination of C to U. Okay, it would there'd be no way to know, is that U really supposed to be there, or was that a deamination of cytosine? Okay, so that's an important thing to point out. Okay. If these are unrepaired, the replication would actually put an A with where there was supposed to be a G. Okay, so if you have cytosine getting turned to a uracil and that doesn't get repaired, and you replicate this, what's going to happen is you're going to have an A for the complementary base of this, instead of what should have been a G, the complementary base of cytosine. The second molecular mechanism of mutagenesis is depurination. And depurination is sort of like the name suggests, it's just a removal of a purine, so that purine base is just cut off. And that can happen from something like a hydrolysis reaction with water. Okay, that water can come in and replace that purine, and now you have an OH group there, and your base free in solution. And this makes what's known as an AP site. The AP stands for apurinic. So apurinic just means there's a purine missing. It doesn't have a purine there. Okay. This happens about 1 out of for every 100,000 nucleotides per day. Okay, that doesn't sound like very much, but there's a lot of bases in DNA. So 1 out of every 100,000 is actually, quite a large number. Okay, it adds up to roughly 10,000 times per cell per day. Okay, it doesn't happen that much in RNA. Okay, this is more of a DNA problem. And again, there's mechanisms in your cell to repair these. Okay. The third mechanism of mutagenesis is radiation-induced mutagenesis. Okay, if you have go out in the sun and you don't wear sunscreen, you get UV light, that induces some changes to your DNA. And those are not very severe, but it changes some bonding patterns in your DNA. And what it does is it creates dimers between adjacent pyrimidine bases in your DNA. Okay. And it's thought that this is probably the main mechanism for skin cancers. We'll show a picture of this here in a second. Okay, some other types of radiation that can create mutagenesis and changes in your DNA are stronger sources like X-rays or gamma rays, and I shouldn't say stronger, I should say higher energy. Right, those cause a little bit different problems, so they're strong enough or high in energy enough to cause rings to open or even strands to break, and those are more difficult to fix. Okay. if you're exposed to, X-rays or gamma rays in a lower amount, those can actually alter your DNA and sort of break it a little bit, but then your cell might not be able to repair it enough, and that can lead to a mutation if that cell divides. So that way, that's why you have, when you go get an X-ray, you tend to put those lead sheets over you, especially in the softer tissue areas that you can have replicating cells. If you're exposed to a much stronger source of X-rays or gamma rays, it can damage your DNA so much that your cell is just like, I can't replicate this. And then it triggers an apoptosis mechanism. And if, you can have this where so many of your cells are just signaling to die and, be removed, that it actually, like, it causes a huge problem. And that's where you have the more acute radiation sickness, where it's not a mutation and cancer that you're worried about. It's actually like you're dying because so many of your cells are being killed off because of that DNA damage. Okay. It's thought that some of these modifications can be repaired. Others will lead to these mutations, and the accumulation of mutation is really what's linked to aging and also cancer formation. So UV radiation, when your DNA absorbs UV radiation, some things that can happen are these bonds between adjacent pyrimidine bases. So they're in the same strand, they're just next to each other in sequence. And these are thiamines, happen to be thiamines, but you have two double bond, carbon-carbon double bonds kind of overlapping one another. When they absorb UV light, you can get the bonds, instead of, this carbon-to-carbon double bond, now you get a bond between these two carbons on adjacent strands. And that's referred to as a 6,4 photoproduct. You can also get a sort of a ring formation between the two, where you have bonds between these two carbons and these two carbons, and that's shown here, and that's referred to as cyclobutane thymine dimer. So thymine dimers and 6,4 photoproducts are the main damage UV radiation causes. And what that does, why that's important, is it puts what's known as a kink in the DNA. So it's kind of that nice B form of the double helix doesn't exist there. There's kind of like a little kink in that spot. And if those aren't repaired, they can lead to mutagenesis when that DNA is replicated. And this is another picture here. There's that cyclobutane dimer that's formed that makes a kink in that DNA. And if the kink is extreme enough, when this is being replicated, the polymerase can't even go through the kinks and actually can stop the polymerase. The fourth mechanism is alkylating agents. And these are agents and chemicals that add groups to your DNA bases. And so if we look at guanine, so we draw guanine, this is the typical way to draw it, but there's also what's known as a tautomer. And for those of you kind of an organic chemistry refresher, a tautomer is an isomer that readily interconverts. And so typically what we're talking about with tautomers is switching of a double bond position. Okay? And these are important in DNA and RNA chemistry. What they do is they change H bond possibilities. and also they change chemical reactivity. So if we're looking at guanine, the guanine tautomer would be where this double bond with the OH moves down, and then this hydrogen migrates to the oxygen. So now we have a double bond between the carbon and nitrogen and an OH group here. That becomes important because something like dimethyl sulfate can then react with this OH group and put a methyl group there. So it would methylate that oxygen. When that happens, now, and I should mention too that this tautomer, this is an equilibrium, these two forms, and the equilibrium greatly favors this form of guanine, the one that we're normally used to seeing. Okay. But there will be a small amount of this tautomer around in DNA, and if you have an agent like dimethyl sulfate there, it will methylate that OH group, and then it forms this, which it can no longer go back to this main form of that isomer of guanine. Okay, so it's changed. This is O6 methyl guanine, and when that happens, then the H bonding characteristics are going to be different. Okay, if we can look at the tautomers of uracil, we have the sort of normal form would be the what's referred to as a lactam with two carbonyl groups. A lactim would have one OH group and one double bond here. You can have a double lactim form of uracil. Now those oxygens are both OH groups, and you've added two more double bonds. So that's just another example of how tautomers are important. Okay, and you can think of that's going to change the chemistry right at these two positions. Okay. Some methylations are purposely done, and one of those here is what's known as SAM or S-adenosyl methionine. Okay, and that's where you have an adenosine group or nucleotide that has methionine attached to it. Okay. These methylations that are purposely done usually occur on adenine and cytosine. They're sequence specific. DNA methylase is a protein that uses adenosyl methionine or SAM as a methyl donor. So this methyl group on SAM is used as a methyl donor to methylate other DNA at specific sequences. And this is used to repair mismatched base pairs. This can be used, this methylation. Okay. E. coli uses methylation as a way to mark its own DNA and destroy non-methylated DNA. So, it marks its own DNA with methyl groups, and then it has enzymes that will chew up and eat non-methylated DNA. So if, it has DNA absorbed from the environment, that DNA will be degraded and not change its genome. Okay. Methylations can affect gene expression, as we mentioned last time, and that term is referred to as epigenetics, where your environment can actually influence your genome. Right. And how this sort of relates is that you have changes in your genome, changes in your gene activity based on your environment, but those changes actually become hereditable. So you can pass those changes on. They're non-base pair changes, so it's not like you're changing, a sequence of DNA. What you're doing is you're changing how that DNA is methylated, and that changes the gene expression levels or the transcription levels of certain genes. Okay, so this is not really a damage per se, it's just really a change to how much a gene or certain genes are expressed, but then that is actually passed on to the progeny. Right. and that occurs again through methylation, either of DNA or the proteins that are associated with DNA in the chromosomes that are called histones. This is currently being studied. There's a lot of research in this because it's a fairly new field, but it's studied as a factor in cancer and possibly obesity. Okay. Causes of this, environmental factors, things that you either ingest or eat, things like nicotine are thought to cause epigenetic changes. Diet, even things in red wine have been proposed to cause these. It's thought that lack of vitamins in pregnant women also can affect this and affect the offspring in sort of negative ways epigenetically, right? So it's not altering the baby's genome, but it's altering how the genes are expressed in the baby. Okay. Which is a little bit kind of scary, right? Like if you're pregnant and you don't take enough vitamins, that can affect your offspring's not just when they're born, but maybe potentially like their entire life if these are sort of not reversible changes. Right. Some other alkylating agents, sodium nitrite, sodium nitrate. Okay, so do those names sound familiar? Those are chemicals that are used as preservatives in things, like food additives. Things like sausages, for instance. These are bad because they accelerate the deamination of bases. And that's one way we've looked at. That's one way that you can induce mutations is by deamination of bases. Okay, they're used as preservatives because they prevent bacteria growth. They don't seem to cause cancer in the amount we eat them, although that is kind of hard to prove because cancer is not something that you can. It requires many different factors to lead to cancer and several mutations. So it's often sort of hard to kind of prove that. But so far, at least in the time that we've been studying these, preservatives, they don't appear to cause cancer. Okay, but the molecular mechanism is sort of there theoretically because they do increase deamination of bases. Right. All of these alkylations play a very small role in DNA damage relative to those caused by reactive oxygen species, and that's the fifth mechanism of DNA damage or mutagenesis is oxidative damage or reactive oxygen species. Okay, so ROS stands for reactive oxygen species. Those are things like superoxide, which is an O2 molecule with an extra electron. So that's what we refer to as O2 dot minus. Okay, there's the minus is a negative charge from that extra electron. And since that electron is unpaired, we usually just put a dot there to signify that. Okay. Other things, the hydroxide ion, OH minus, and hydrogen peroxide as well is a reactive oxygen species. So if these are around in your cell, that's not good because they can actually react with your DNA molecules and damage them. Cells control levels of these with enzymes called catalase and superoxide dismutase. So those enzymes are specifically to remove these reactive oxygen species. Okay, lots of reactions can occur with these reactive oxygen species. You can oxidize your deoxyribose sugar, you can oxidize your bases, you can actually cause strands to break. So there's many, it's not really like in terms when we looked at deamination, there were some very specific reactions that occur in that deamination process. But with reactive oxygen species, it's not really the specific things. It's just, it can react so many different places in your DNA. Right? And so with that as well, it's kind of hard to have a cellular mechanism to repair that DNA once those things happen, because there's so many different places for those superoxide or reactive oxygen species to react on your DNA. Okay. And this figure just kind of shows you where these reactive oxygen species come from. Okay, they come from your main electron transport chain. They can form. So superoxide can be formed in complex 1, complex 2, complex 3. Okay, and that's where superoxide dismutase comes into effect. It can react with superoxide and form hydrogen peroxide. That is also bad, but that's where we have another protein, catalase and GPX, that turns that from hydrogen peroxide into water, and water is really benign. Okay, so that's how your body can sort of pacify these reactive oxygen species, and these are always forming in a small amount when you're using the electron transport chain, which is all the time. Right? DNA is the only macromolecule that has the benefit of biochemical repair systems. Okay, and that'll be discussed later in later chapters. But lipids, if they're damaged by these superoxide or reactive oxygen species, they're not going to be able to be repaired. And likewise with proteins, they don't get repaired, but your DNA does. Okay. So the last thing we'll talk about in chapter 8 is the small section, and that's just other functions of nucleotides. And some of these we've seen before. Some of them you've probably seen before in other classes. Okay, the first other function of nucleotides, other than being in having to do with storage of information, would be as an energy source. And the most common one that we refer to for that is ATP, right? ATP, we have our ribose base and 3 phosphate groups attached to that at the 5' position, and then we have an adenine base on that. So that would be our nucleotide. And the triphosphate group is rather has a lot of potential energy there in it, because you have so many negative charges so close together. And losing that gamma phosphate group, removing that, really releases quite a lot of energy, and you can couple that release of energy to a reaction that is not favorable and actually drive that reaction forward. Okay. Another function of nucleotides are as coenzymes. And we've talked about these before as well, because we've talked about in terms of enzymes, some of them use what are known as coenzymes to help catalyze their reactions. So one example of a nucleotide that's used as a coenzyme is the NAD or NADP, nicotinamide adenine dinucleotide, right? And so our nucleotide part is down here, right? We have the adenine and ribose sugar and the 2 phosphate groups, and then that's attached to another ribose and the nicotinamide part, which is actually the part that can accept electrons. Okay. And flavin adenine dinucleotide is the same thing. We have a riboflavin group up here that can accept electrons, and then here is our shaded, would be our adenine nucleotide. Right, and then finally, our coenzyme A, and I think I've mentioned this before as well. Really, the coenzyme A, what you're looking at here is the SH group, because that can attach to some things and connect to them and be kind of a carrier, but our nucleotide parts over here, where we have adenine and our ribose sugar and a diphosphate. So really these, when they're, the nucleotide part of these cofactors is really kind of just like a building block. The ADP part of all of these is like a building block that you put something else on that then has a biological function. Right. And the example of where these coenzymes become important, one example is in the enzyme system pyruvate dehydrogenase. So pyruvate dehydrogenase, and you'll cover this next semester in metabolism. But what it does is it converts pyruvate into acetyl-CoA. Right? And how it does that is by using cofactors, and it uses quite a bit of cofactors. TPP, FAD, lipoic acid, NAD+, and coenzyme A. And so three of those we saw on the previous slide, the FAD, NAD+, and coenzyme A, we kind of saw there on that previous slide that contain nucleotides. And just briefly, the coenzyme A comes in, pyruvate dehydrogenase takes off a CO2 from pyruvate, and then it sticks the acetyl group that's left over onto coenzyme A, and that gives you acetyl-CoA. Right, CoA just stands for coenzyme A. So you have an acetyl group attached via that sulfur group on coenzyme A. Right? And to do that, this complex, pyruvate dehydrogenase, will use an NAD and convert it to NADH, which is the reduced form. Okay, it gives off CO2. And then it needs to, if it's going to keep doing that reaction, it needs the NAD to be converted back to NAD+, and that's where it uses the FAD. The FAD will take electrons from NADH and then transfer those electrons to the main respiratory chain. Okay, so that's just a little preview of metabolism there. Okay. The third other function of nucleotides that we'll talk about are in regulatory molecules. And so we've seen two of these before, and one really specifically when we talked about cell signaling, and that was cyclic AMP. So in the real name for this, the full name is adenosine 3'5' cyclic monophosphate. So you have an AMP where that phosphate group then forms another bond with the 3' oxygen and gives you kind of a ring form here. That's why we call it cyclic AMP. And remember, cyclic AMP has many different roles and different signaling pathways. Okay. We also saw in that chapter 12, we saw a mention of cyclic GMP in the rhodopsin pathway. So that's another regulatory molecule very closely related to cyclic AMP. You just have a guanine base. And then this molecule here, guanine 5' diphosphate and 3' diphosphate, is another regulatory molecule that I don't believe we're going to see a specific example from that one. Okay, so that really wraps up our discussion of nucleic acids. The next lecture and final lecture of the semester will be covering chapter 9, at least the beginning of chapter 9, talking about some DNA-based technologies.